My name is Heikki Seppä, and my title is indicated here. It's a little different than I originally indicated. Now I call it Rapid Climatic and Biodiversity Turnover Rates. And there are lots of people included in this work. As you will see, it's based on fairly substantial amount of data. It would be impossible, at least nearly impossible, for one person to create so much data. If somebody can do it, I would like to meet that person one day. Anyway, here is background for my talk. We all know that we are living in rapid climatic change, and it is, of course, possible to calculate the current rate of temperature change. It has been done in a number of studies. This is studies published 2 years ago by Smith et al. And in this study, they calculate the rate of temperature change for different parts of the world. And if you look at the northern hemisphere, the rate of warming has been roughly 0.2 degrees per decade. So in one sense, this would be, of course, a 2-degree warming. Okay. We also know, based on very many studies, that associated with this rapid warming is right now taking place a massive reorganization of biotic communities, including terrestrial, aquatic, and marine communities. An eventual outcome of this is very likely what some people call novel ecosystems or novel communities. Then the question I have here, and what's the basis of this presentation, is that how can we use paleo records to investigate past rates of temperature change and associated biotic turnover rates. In my talk, I will focus on what we call the late glacial. We can also call it the Pleistocene Holocene transition from roughly 14K to 11K. As we all know, at least almost all of us know here, it was a time of rapid climatic changes, including both rapid warmings and rapid coolings. We have lots of evidence for that. A few years ago, we published this paper where it was first authored by my colleague Heikki, where late glacial temperature reconstructions were presented for different parts of Europe. These are all based on kilometer data. It's a summer temperature reconstruction. And I will just pick one region here because I will focus on this. It's the Baltic region, but in this paper, for different sectors of Europe, a composite summer temperature reconstruction was presented. Now, based on this data, which is also indicated here, we have a late glacial. This is what we call Berlin-Allerrod, or last interstadial. Then we have the stadial Younger Dryas. We see the summer temperature, and this is the Younger Dryas Holocene transition. Based on this data, we can then calculate the rate of temperature change and compare this rate of temperature change with the modern rate of temperature change. So here we can see that these bars indicate the temperature rate of change. The scale is here. This is per year, warming per year. You can multiply by 10 if you want to have it per decade. So we can see that at the beginning of Younger Dryas, the rate of temperature change was over 0.3 degrees per decade. And at the Holocene, Younger Dryas Holocene transition, then indicated here, the highest rate of temperature change was 0.17 degrees per decade. So it's fast, but as a matter of fact, it is a little slower than the current Northern Hemisphere mean rate of temperature change, which is 0.2, as I said earlier. Okay, this is about the rate of temperature change. Then, as I said, we want to study how the biotic communities responded to these rapid climatic changes. This is where some new solutions had to be done, as I will explain in a minute. The background is in modern ecology, modern plant ecology, especially work by a very famous scientist, ecologist Robert Whittaker in the USA in the 1960s. And he defined this concept of biotic community turnover. Nowadays, many ecologists call this beta diversity, especially when we are dealing with spatial diversity. When we are dealing with paleoecological data, it's a little different than spatial diversity. Obviously, we are mostly looking at temporal change of communities, because we are working in time. In this case, if the samples are temporarily arranged from the oldest to youngest, or vice versa. They then are directional in relation to time. Here in this study published by Anderson Dahl, which is a review of beta diversity, we can see the idea of having a directional change in communities. So here it can be a spatial gradient, but it also can be a temporal gradient, and we have a samples changing in time. In our case, the samples can be, for example, time bins, let's say 300-year bins or 500-year bins. However, we have to keep in mind that although it's directional to time, it's not directional to temperature, because as you know, in late glacial, sometimes the climate was getting warmer, but it was also getting colder. And that has some implications. Another big issue is whether we are dealing with presence-absence data or quantitative data, which is abundance data. And there are lots of articles discussing which is the better way to measure the compositional change in past samples when we're dealing with fossil data. In this study, for plants, when we have macrofossil data, we take it as presence-absence data. Those of you who have done work with macrofossils probably know that it's not really absence data. Absence of macrofossil doesn't prove really absence, but it's presence data anyway. We can take it that way. Pollen data is more quantitative, theoretically okay, but we have some problems with pollen data as well, because the source area, especially in the late glacial pollen samples, is vaguely known. We can have a pine pollen value of 3%. Where was the pine growing? Who knows? So we combine both plant macrofossil and pollen data in the study. Also, there are very interesting animal fossil records, for example, mammoths. So there are some bone-based mammoth records available. And also, there would be records for aquatic ecosystems based on aquatic microfossils. I have no time to discuss aquatic communities in this presentation, unfortunately. And we have to focus on one region, because any region we want to study this way needs to have a record of pollen, macrofossil, and other data. So there must be a fairly dense network of records. Also, what we had to do in the study, after some trials and errors, was we had to pool all plant macrofossil records into one record. So what we did, we took 8 plant macrofossil records and made one composite plant macrofossil record for the whole region. And it looks like this. This is not very often done in paleoecology. I'm still thinking about it, whether it's okay or not, but I don't have a major problem with that myself. You can correct me if I'm wrong. Paper has been published. So pollen data is different. It's obviously quantitative. Also here we binned 500 bins, and for each bin we calculated the total turnover for the bins using tetraentity canonical correspondence. There are also other ways to assess the turnover with pollen data, which is, as I said, abundance data. Okay, here then results, and all the results are here in one composite output. First, this is the rate of temperature change, as I showed earlier. The bars indicate how quickly the temperature was changing. Either it was getting colder, or then it was getting warmer, as it was getting warmer in the Younger Dryas Holocene transition. This is the pollen turnover, and it's highest here in the Younger Dryas Holocene transition. It's quite low in the Holocene. Holocene is quite stable. And also the Berlin-Alleröd interstadial is quite stable. So the turnover is clearly highest when the temperature is changing, either getting colder or getting warmer. Aquatic turnover quickly. Again, we can see that the Younger Dryas, very stable communities, and higher turnover, for example, at the Younger Dryas Holocene transition. Plant macrofossil as a record of presence-absence of plants. Here, clearly the highest turnover based on CNRS index is here when we have the Younger Dryas Holocene transition again. Then we also mapped the key regional extinctions, because they are part of the turnover process as well, for Arctic alpine plants in this part of Europe and also for some glacial mammal species. So, for example, Salix polaris, if you look at the macrofossil record, it is present all through the study period and goes extinct here at the end of Younger Dryas. Saxifraga isoideus, another Arctic alpine species, also present until the end of Younger Dryas. And finally, the most famous species of late glacial, I think, Dryas itself. This is Dryas octopetala, fairly common both in pollen samples and plant macrofossils, as you can see here, until it goes extinct at the end of Younger Dryas. Regionally extinct, of course, not globally extinct. Also, we have some, as I said, some mammal species, which we can study from the same way as well. This is reindeer, now regionally extinct. This part of Europe was present until the end of Younger Dryas, and as a matter of fact, in the earliest Holocene as well. And finally, another famous late glacial species, mammoth, was present also until the end of Younger Dryas and goes here extinct. And this is actually the final regional extinction of mammoth in Western Europe, the youngest radiocarbon dated mammoth in Western Europe. Okay, as we can clearly see, the most distinct compositional turnover in these different components seems to take place at the Younger Dryas Holocene transition, both in pollen, plant macrofossils, and in terms of regional extinctions. So that's the key transition period in the Pleistocene Holocene transition, at least in this part of Europe. So my conclusions: most distinct biotic turnover takes place at the Younger Dryas Holocene transition when the Earth, or this part of Europe at least, was getting warmer with the rate of 0.17 degrees per decade, which is almost the same as the current warming rate, little slower. Associated with this general species composition shift were regional extinctions, very conspicuous regional extinctions, including, for example, mammoths and many, many Arctic alpine plant species. So in this way, we can see especially the Younger Dryas Holocene transition as an analog to the situation what we are having right now, especially in the subarctic areas, where the summer temperature now is roughly comparable with the summer temperature during the late Pleistocene in many parts of Europe. Thank you.
I have a question about calculate the turnover, because if you have increasing in diversity, do you expect an increasing in diversity during a transition from one community to another? So this how affect the calculation of the Southern index? Because higher number of species, you increase the index, but it's just a change on the diversity.
Yes. You mean like yeah, you probably mean richness, like richness more than diversity. Yes.
Well, richness, yeah.
Richness easily could be calculated here as well and shown here as well. We may have done it as well. I just don't remember off the top of my head whether we have a richness curve as well. I see your point, of course. Yeah. Okay. Yeah.
Thank you, Heikki, for setting the scene so well. So my focus is going to be exclusively on the vertebrate record, and specifically actually on the mammals, and also covering the same time period, but this time from Britain. So I think for many of us who have been engaged in vertebrate paleontology, there's been a real issue for us over decades now about the nature of late glacial responses, particularly, of course, the extent and the speed of those biogeographic shifts in range, but also the impact of those climatic oscillations on extinction events. And in fact, actually, of course, one of the things that comes out constantly in the literature is this discussion of disharmonious faunas. And I find disharmonious a strange term anyway, because it's has a very presentist feel to it. So anything that looks different to what we're in now is described as disharmonious. But the real issue for us has, of course, been the lack of completeness in terms of a record that we can examine. And we have been really very much the poor relation compared to the types of studies that Heikki has shown, particularly for pollen or chironomids, for example. So lack of chronological control, lack of stratigraphic control, and lack of resolution with the types of sites that we've had. Now, for Britain, we've also been, we're in a privileged position in terms of sensitivity to the North Atlantic and potential impacts on our mammals. What you can see here is actually a record, a composite record drawn together by Somme and colleagues from sites in southern Scandinavia, which show, in fact, that when you have an obligate cold climate species, such as reindeer, and extending into the Holocene, as we've seen, an obligate thermophile, which is our pond terrapin, Emys orbicularis, that actually, in southern Scandinavia at least, there is no overlap. There's only a few hundred years between them, but there is no overlap between them. Now, in fact, as you move further south, the picture is substantially blurred, and that's been one of our issues in Britain. Are we really seeing disharmonious faunas, or is it complicated by our lack of resolution? So there's been a big need for better sites and better dating. Now, the sites that I'm going to discuss in the context of the present talk are broadly located in the southwest of England. They're in a limestone area and comprise a number of cave sites, but I'll also be referring to a number of cave sites in the Creswell Crags area in the East Midlands, and then finally an open site with reference to extinctions in the West Midlands. But the real focus is going to be on these sites down here, which are scattered up and down the limestone area of the Mendip Hills, and in particular this new site at Gully Cave in Ebor Gorge, which is located just outside the cathedral city of Wells. So here's the cave, perched high up on the side of a wooded limestone gorge. We've been working there for 10 years. It initially was completely unexplored, just a small entrance here, and over the last decade we've expanded it substantially. Now, in fact, this cave preserves what seems to be a pretty much complete sequence, at least going back, we now know, to stage 3, we're still going down, and finishing up in the early Holocene. The deposits consist of a series of limestone breccias. They are largely horizontally bedded, undisturbed, and they are capped off at the top by an as yet undated but mid-Holocene, I'm almost certain, flowstone floor. You can see it going around the deposits here. We've excavated over 100 metric tons of sediment, pretty much all of which has gone through a 500 micron sieve to extract small vertebrate remains, but there's also large vertebrate remains as well. There's not only mammals, but also birds, reptiles, amphibians, fish, and there is a reasonably respectable molluscan record in there as well. We've now got over 11,000 vertebrate remains, so it does provide a reasonable degree of confidence to us in terms of the types of information that we're able to extract. Now, I'm just going to talk about the upper part of the deposits. We do have a sterile, or almost sterile, sequence of last glacial maximum deposits below, and then stage 3 deposits below that. But I'm just going to talk about the late glacial material through to the early Holocene. And you can see here from the spread of radiocarbon dates that we have that we have both the early Burling, Alleröd, Younger Dryas, and indeed the Holocene, at least the early part of it, recorded in the cave before the cave is pretty much infilled, inaccessible, and is capped off by the flowstone floor. I think important points to note at this point are extreme diversity, which we'll get onto in a moment with the late glacial interstadial. Certainly our maximum diversity occurs during the Burling period. To note also that by the time we get into the Alleröd, we're picking up more abundant remains of red deer, Cervus elaphus, and in fact, actually, a complete absence of horse, Equus ferus, at this time. This is important. I'll return to that later. And then, of course, a good Younger Dryas and into the early part of the Holocene. So the rest of my talk is really structured along the lines of the title of the session, Do Species Move, Adapt, or Die? So if we come on to the do species move, our answer is unequivocally yes. So certainly the information that we have for Gully Cave is that we have shifting percentages of taxa, some of which I've outlined in red, and I'm focusing here specifically on the small mammals. They are the most abundant of the groups. We know that they are at least locally derived, brought in by birds of prey. And I've colour-coded them according to species that we would find today living in this part of the southwest. So predominantly several species of vole, Microtus agrestis, the water vole, Vicola terrestris, the bank vole, the mice, and the saurissids, Saurox minutus arenaeus, and the water shrew Neomisphodians. Other taxa, such as Lepus timidus, the mountain hare, and Microtus arvalis, the common vole, are today located outside this part of southern Britain. They're predominantly in the north, up in Scotland or in Ireland. And then finally a suite of cold climate, more exotic taxa occurring in the interstadial. And some of these are really, they are far movers. So this is a modern distribution map of one of the tundra voles, Microtus gregalis, but co-occurring with other species of Microtus and a couple of species of lemming. So certainly at this point we have enormous evidence for the reorganisation of communities, different species coming in. And of course that movement is facilitated by the fact that Britain is connected as a peninsula to Europe at this time. So this map shows various coastal reconstructions going all the way from the last glacial maximum into the Holocene. Now, of course, you still have the Channel River here, which would have acted as a significant biogeographic barrier, but nevertheless, the access route from the continent is clear. And we can see, certainly from the chronology, when we compare late glacial sites across Britain, that these southern sites are occupied first and then a progressive movement towards the north. The most extreme evidence of movement, however, comes from this species here. This is the Saiga antelope, Saiga tatarica, today a Central Asian species. And you can see from various sites in the Mendip Hills, those southwest sites, that we have an extraordinary incursion of this species, a massive westward movement at this time, all underpinned by good recent ultrafiltered radiocarbon dates. Now, in terms of hominin movement, the late glacial marks the return to Britain of hominins, of course Homo sapiens in this case, and they are there right from the word go on the rise, the rapid warming of Burling. The sites that we find earliest are these ones down in the southwest, and there is an intense focused predation on horse. You can see here there's some teeth. The cut marks on the teeth haven't come out too well. These are from one of the sites at Goff's Cave in Cheddar. But nevertheless, this makes an important point, underlying the fact that the Burling is not the most wooded part of the late glacial interstadial. In fact, there's a lot of open ground conditions here which support large numbers of horse. Slightly later, we start to see occupation of those midland sites, actually technological changes as well. An important difference because the transition from the Burling to the Allerød sees the disappearance of horse across large parts of southern Britain in response to the spread of woodland dominated by betula. And in fact, there is a switch from horse-based predation. You can see some horse teeth here that actually have been smashed open for the extraction of bone marrow, and a focus towards intense predation on Lepus timidus, the mountain hare. In the Younger Dryas, the evidence from Galley Cave has given us our unique stratified Younger Dryas site in Britain, because otherwise our evidence had previously been drawn from isolated finds. But here we have well-stratified material. And I've just put in some of the small mammals here, and these have all come out of the same level, in fact, actually out of the same cluster of raptor pellets. And you can see, for example, that we have huge differences in biogeographic range. So, for example, animals such as the steppe pika, this leporid, Ocotona pusilla, with an Eastern European steppic distribution today. But to draw your attention to the fact that we have three coexisting species of lemming, Dicrostonyx, the collared lemming, Lemmus, the Norway lemming, and Ligurus, the steppe lemming. This is only the second record of this species in Britain. All of these co-occurring, but today not sympatric. And at this time, the cave appears to have been a den predominantly occupied by Arctic fox, so Vulpes lagopus. Do they adapt? We have no evidence for this at the present time. Certainly mammals are very subject to potential size change. and morphological change, but there is no consistent pattern for this. Now, this may well be because there is insufficient time because of the rapidity of climate change. The need to adapt is being offset by the fact that the animals are highly mobile. But it could also be buffered by complex topography, the fact that these sites occur in steep limestone valleys in many cases, where there would have been a microclimate. And this draws back on some of the previous work that's been done by John Stewart and Adrian Lister a number of years ago, which make the point that actually that the need to adapt is offset by the fact that these act as refugial areas. And you can see, in fact, that actually, if we look at the Younger Dryas levels in Gully Cave, this is just a 10 centimetre spit. In the blue, there are predominantly the cold-adapted taxa, but there is still a substantial proportion of species that require wooded or rather more temperate conditions. And in fact, actually, as we get into our highest Holocene levels, in fact, we see our collared lemmings and Norway lemmings hanging on here, but a large change in terms of overall composition of the fauna. Do they die? Yes, without doubt. So we saw that Mammuthus was last noted in the Burling in Britain, obviously persists a little longer in Scandinavia. We see the decline of Equus during the Allerød. Is that a local extinction or are they moving? We don't know this at the moment. But there's probably a lot more cryptic turnover than we ever expected. And we can see that actually in the DNA. This is a really important paper that was published a few years ago by Selina Brace and colleagues, demonstrating in Dicrostonyx torquatus, using mitochondrial DNA, that this is not a single species that is persisting through, in fact, going back in time into stage 3. But in fact, these are different, distinct genetic clades. So there's probably a lot of cryptic extinctions, local extinctions that we're not seeing. And finally, of course, some persist longer than we expect. So things like Microtus arvalis, we've been able to demonstrate through a combination of geometric morphometrics and ancient DNA that this species, thought to go extinct at the Holocene boundary, is in fact persisting into the Holocene. So just to wrap up, we now can see that with these new archives, we can use a battery of new techniques, a high-resolution approach, to underline the reality of these disharmonious faunas and potentially the operation of this area as a cryptic northern refugium. And of course, this provides not only the paleo-environmental backdrop to Homo sapiens, but also important paleo-dietary contributions in terms of the types of species that are available for exploitation. Thank you.
It's very beautiful data and, by the way, very clear talk. Thank you very much. Can we exclude at these times and timescales effects of human predation?
In this particular cave, I think yes, because for the majority of the material that I'm discussing is microfauna. We do have some larger mammals as well, but there is no clear evidence. There's no artifacts at this particular point, and there's no clear evidence of hominin use. There are some materials that appear to have been broken open for marrow extraction, but they occur only during the burling. This cave isn't being occupied by hominins. They tend to mess things up.
It's diversity, as the session says. It's Europe as we have been, but we're sort of moving to pollen, while Heikki talked about pollen, I suppose. This work draws on the European pollen database and basically the work of Steffen Wolter, Jacqueline von Leyen, and Pim van der Knaap, and Simon Brewer did something as well, I believe. The task behind was to look at the taxonomy in the EPD and to unite them, and this is why these three people were instrumental, and I wouldn't have been able to do anything without them. The whole work wouldn't be anything without many people in this room that have contributed to the European database, which we gratefully acknowledge. So this is a huge community effort, which I have the pleasure to present. So the latitudinal diversity gradient is a sort of pervasive pattern in space, a diversity pattern in space that was already recognized by Alexander von Humboldt. So it's a big thing, everyone sees it, it's there in most biota. So we have most species at around the equator, and then it sort of drops off. If we look at plants, it looks a bit more patchy than these. Then we all know that in the dry Sahara there are not so many species, so it's sort of a more difficult pattern. But in general, you see the high diversity areas are near the equator, and if we go to the pole, we have fewer plant species. This pattern was tried to explain by many hypotheses, modeling scenarios, sort of addressing these and trying to explain it statistically with just regression models. In these regression models, one parameter is often in there, and that is some sort of area. In this case, it's potential evaporative transpiration. Sorry, not area, energy. So temperature seems to be one important factor that can explain a bit of this latitudinal diversity gradient, but not all of it. But if we think that temperature is important, and if we think that we had sort of movements within the Quaternary or just the last 15,000 years, and we always know that we know that our sort of trees move north, as in this example, Quercus in Europe, so that we think they were somewhere down in the south when it was cold, and then it gets warmer, they all move north. And we sort of apply these concepts automatically to most of our other taxa, and sort of think that, well, everything was down south, and then it gets warm, and everyone moves north. If we would assume this concept really, that would mean that our diversity gradient in Europe should have changed from the late glacial into the Holocene. We can basically hypothesize that the post-glacial warming should have affected the latitudinal diversity gradient in Europe. As species spread north, the gradient should have become flatter between the Mediterranean and the boreal zones. So before, in the glacial, it should have been very steep. Everyone should have been in the Mediterranean. And north of it should have been sort of really cold and no one wanted to be there. And then it gets warm and everyone spreads north, so the gradient should sort of flatten down. Conceptually, this would be the concept. So let's see if we can see that with the data that is available to us. We still have to solve a bit of a problem, and that was mainly the work of Stefan Wolters and Jacqueline and Pim. And that is, in Europe, we have different pollen floras. Different pollen floras call the same thing different names. As in this example, Helianthus annuus, in the three most commonly used textbooks in Europe, it sort of keys out a different thing. So we need a synonyms table before we start anything. Otherwise, we would sort of create diversity where there is none. This is then the surface sample data of data sets in the EPD. So we still see we have sort of some funny peaks. So there is something else that we need to worry about. And that is that there are some people that are really good pollen morphologists, and they can identify these things very well and notice at which latitude this sort of peak occurs. This is southern Germany and the Alps, and there are some very good pollen morphologists working there. Now, the problem can be illustrated as such. This is a slide that I stole from Stefan Wolters. So he would say, well, someone with a long experience, plenty of time dedicated to pollen morphology, such as Jacqueline, I suppose, and a substantial reference collection could sort of identify all these different Apiaceae pollen, whereas someone like me that doesn't have lots of time would just call them Apiaceae. And that creates a little bit of a problem, so that we try to solve these by creating a hierarchy and sort of thinking about making a morphological hierarchy, thinking about how would sort of people with different skills sort of identify these pollen grains. So we have the accepted EPD name. The list was initially around about 4,000. It was brought down to 1,200 accepted names. And then if we look at the second higher level, which we find useful, we're down at 400 sort of pollen types that are recognizable. And that sort of fits with our European palynological textbooks, which recognize that sort of many types or a bit more. Okay, so this is the data. We're jumping right into the results. I have to explain I did something more with these. So I used all the pollen diagrams in the EPD that had at least 30 identified taxa and some sort of chronology that was useful. I binned all these samples into 1,000-year bins in order to create larger samples, because we will mainly be looking at herbaceous pollen types, which are much more diverse than tree pollen types. And so we want large pollen sums. So we're looking at big pollen sums. Still, the number of pollen types counted is the best predictor for the number of pollen types that someone will be able to identify. There is a very good correlation. So therefore, if we do any sort of comparisons, we should always do rarefaction analysis. So these means that these data here is verified, all the SIG lines are verified to 500, but also the dotted lines looked at what is if I don't verify anything and just look at it. So we're looking at 1,000-year samples, and we're looking at the accepted variable and the second hierarchy. We observe that our R square, it means that we predict the way our regression model sort of explains our cloud. So our dots are a little bit tighter around our regression model for the hierarchy 2, which is expected. So therefore, we get a little bit higher R square than just for the accepted, but not so much. And the slope is, of course, reversed for these two. What we do see, and with the raw data, if we do the same with the raw data, we find similar trends in the R squared and also in the slope. And this trend was surprising, and it was opposite to what we would have sort of hypothesized. We have a very, very sort of shallow gradient in the late glacial, and that gradient is then steepening as we go into the Holocene. And it is actually steepening if we compare that to the North Grip. If we just scale the North Grip to the same sort of 1,000-year intervals, we even see a sort of blip of the younger dryas in our gradient here, and we see a rise which is at least 3,000 years delayed after the warming. So that was a sort of surprise. I went one step further and then sort of pooled all the pollen data. So all the samples that I had sort of pooled into 1,000-year bins, I now pooled them together into just one big sample per each region. And I was very pragmatic and simplistic, and I just decided I want a boreal region. I included Scotland and sort of cut it off in southern Sweden. And then I created 2 temperate regions, one oceanic, one continental, and a sort of extended Mediterranean, sub-Mediterranean type region. I took out the Alps because they're a bit special, partly due to the Swiss people having done a lot of work. These are the results if we look at these pooled samples. First of all, it's really nice to see we always have the highest number of pollen types in the Mediterranean. Again, these samples are now verified to 50,000. So again, we still have to do rarefaction if we do want to make sensible comparisons. So it's nice to see the Mediterranean samples are always richer, or the region, and the boreal is the poorest, and the other 3 regions lie in between. Then, first of all, you see an obvious trend, and that is round about at 5,000 years ago. These sort of temperate regions, they go up in pollen type richness, and that can be explained with the action of humans in Europe. We are starting to open the landscape. We are bringing in species. The other trend that was not so obvious and not so expected is while our Mediterranean sort of region doesn't show a very big trend coming from the late glacial into the Holocene, the boreal region shows a decline, as does the continental temperate region. And this is an effect similar to what we just heard of the forest coming in and sort of closing up everything. And so basically this is our early Holocene refugia period for many of the herbaceous species, which are outcompeted by forests coming in, covering everything, and they sort of survive the early Holocene in special habitats until we open up the forest, and then the same sort of type of species come up again. So if we now do just the difference between these regions, so this is the curves for the difference between the Mediterranean to the boreal and the 2 temperate regions, we see that the difference doesn't really change in the early Holocene, but it does in the late Holocene when we sort of bring in species. So what we were postulating should have happened in the early Holocene, we do see in the late Holocene with the opening of the forest. So the reason for the difference in the trends of the bigger regions and individual sites may be explained a little bit with this analysis. This is an analysis of looking at how different are the pollen compositions at the different sites. And that is done by the taxon accumulation curve and measuring the slope. So they are more different in the late glacial and the early Holocene, and then they become more unified, more similar to each other. So the overall pollen taxonomy or pollen taxa number in the region stays the same. Individual sites get more taxa, but this is because processes that makes the landscape more homogeneous. So with that, I can conclude. I think that fossil pollen data can provide some insight into continental changes of vascular plant diversities. Even so, there are many different difficulties relating pollen to plant diversity, which I do not want to sweep under the carpet. I think we see competitive exclusion, which has lowered the regional pollen pool of types of the temperate and the continental temperate and the boreal regions, with the post-glacial spread of the forest, and thereby they are steepening. This is the effect which is contributing to steepening this gradient until 7,000 years ago. And we clearly see in Europe the effect of land use, which has affected the latitudinal diversity gradient over the last 3,000 years and actually flattened the gradient between the regional pools, which we had expected to occur with the beginning of the Holocene warming. Thank you.
I like the last slide. You talk about this homogenization of the taxa. So, and this happened before human intervention. So this is a natural way. You expect the nature evolved to homogenize without the intervention of humans?
Yes.
So
the trend started before.
Yeah. Well, because this is a question right now, no? Is the humans homogenize the Earth or not?
Well,
it seems like this is a natural way if you don't have a big change on climate or whatever.
The mechanism that I see there is that in the early Holocene, plants did have to go around. Some plants may have survived in cryptic refugia, but if they did in cryptic refugia, these were tiny pockets. And out of these tiny pockets, they did have to sort of spread over the landscape. Also, some plants did have to move and did migrate, and trees, for once, we have very well established that. And those often did that with long-distance dispersal jumps. And they made a long-distance dispersal jump, and then out of this jump, they then had to colonize the area. So we have, in the beginning of the Holocene, probably pockets which are more different in vegetation composition. Same even probably happened in the Mediterranean. The Mediterranean during the last glacial maximum was a dry area. So all our pretty trees and flowers, or many of them, were probably in sort of small pockets, mountain ranges, and they did spread out, because today we don't. We have an often forested area. So these are natural processes, yes.
And the derivative of that is always there is a space for a new species to arrive. So it's not saturated, the communities are not saturated.
They were not saturated, although there is probably a mass effect which doesn't allow other species to come in. At least we can sort of see that for the trees.
Okay, cheers, man.
So my purpose is to go to see what snails can tell us about, well, mollusks, better than snails, can tell us about past biodiversity. And to do that, the Somme Valley is a very interesting region because it has been investigated since the end of the 19th century, because it's where prehistory was born, as many of you might know. And mollusks have been the subject of attention since the very first studies undertaken in this area, until the last 20 years, where the record have been very much increased. So there are many information available through the literature and many new data. The interesting thing of the Somme Valley is that there is a very well-stepped terrace system. This is the work of Pierre-Antoine, one of my co-authors. And the point is that this terrace system valley is formed by 10 alluvial formations, and Pierre-Antoine demonstrated that this is linked to climatic cyclicity, each step being corresponding to one climatic cycles with coarse sediments during glacial times and fine sedimentation during interglacials. So the 34 localities that have yielded mollusks in the Somme Valley are distributed within this terrace system, and they cover a range of time of 1 million years. So this chronology is available through many datings obtained by OSM method or luminescence method, OSL, or uranium thorium, or for the most recent part through the radiocarbon dates. Chronology is also reinforced by a new method developed by Kirsty Penkman, who is also co-author of this talk, and which is an amino acid chronology. Kirsty works on the opercula of Bithynia shells, which are quite abundant through the sediments of the Somme Valley system. And on this figure, you can see that all the results from the Somme Valley that I have highlighted in red shows that the records obtained by this amino acid chronology is very consistent with the terrace system and is also consistent with the results obtained in the Thames Valley, which is plotted here on the right side of the figure. So this gives very much consistency to the dating of this alluvial formation. So let's go to the different sites studied for mollusks, of course, are not homogeneous in terms of sampling. And on these schemes, I have plotted in red the number of sites for each of this period. And I have plotted in blue also the number of samples, because some periods, like 5E, for example, might be represented by only one site, but a very good site, which is Kaur here, and which yield very, very many mollusk community and a long story about the evolution of this mollusk community throughout the late glacial, the interglacial, pardon. While during MIS 9, for example, you have also only one site, but there is a very poor record because this is from an old publication. Along this 1 million year record, only 2 hiatuses have been registered affecting the Cromerian period, but on the other part of the record, there is a continuous information. So first, if we are looking at the number of species, because most of mollusks can be identified at the species level, then the first thing to see is that temperate phase, of course, exhibit the higher biodiversity, that cold phases yield less than 20 terrestrial species, and here we have the effect of a bad sampling just for this interglacial period. Biodiversity of land snails are interesting also to consider extracting the forest species, because the forests are the most diversified group among snails. So here the curves show that when you have more than 30 species and a high percentage of forest species, you are always in interglacial periods. Between 30 to 20 species, then you are in interstadial periods, but as you might note, not all the interstadial include forest species. And when you have less than 20 species, then you are almost during cold phases where there are no forest species. So the other step I wanted to look was how species are distributed within a climatic cycle. Let's say, so of course interglacials are producing the higher numbers of species, and they are distributing within quite different ecological groups, while of course glacial, pleniglacial faunas are quite poor and distributed over a limited number of ecological groups. What's interesting also to consider are the transitional phases, early glacial and late glacial, that are giving almost the same numbers, except the fact that during early glacial, forest species are still present in the malacophonas. The second observation refers to what I've called exogenous species. So by exogenous species, I'm including extinct species, but I mean real extinction, so species that do not exist anymore anywhere, and species that occur in the Somme Valley, but that are out of their modern range. In this came, of course, the higher rate of exogenous species is in the interglacial periods, and it's also the only climatic phase exhibiting some extinct species. Again, the lower numbers are for pleniglacial, while the transitional phases are giving more or less the same proportion of exogenous species. So the following question concerns what are these extinct species, and do they have a chronological range, and can we define some specific faunas for particular periods, and especially for interglacial periods? So first, extinction in the Somme Valley, I was able to identify 4 species that become completely extinct. And because it's quite a narrow area, I had a look also to the Paris Basin around and identified 10 extinction of species for the same range of time, and 8 in the UK. And in the total, in this area, including Paris Basin to the UK, we count 13 extinction of species of mollusks, but no extinction occurring after stage 7. If I look now to the species out of range, then it's clear that the periods that are the richer with this category of species are Middle Pleistocene interglacials. There are also quite some of those during the Holocene, but there is a strong difference between the Holocene and the Middle Pleistocene interglacials. So the question, of course, the following question is, what are the missing species in the Holocene? So on this table are listed all the species that are absent in the current fauna of the Somme Valley, but that have been registered in some quaternary deposit of the same area. Here, you have the occurrence during the Holocene, the Pleistocene interglacial, interstadial, and stadials, and these notations refer to the modern range of species, so Western species, Northern European, Central European, Alpine, and Southern species. So what this table is telling us is that first, there are 32 species that have been registered during the Quaternary in the Somme Valley and that are not existing anymore. And among these 32 species, 5 were recovered during the Holocene, but the 27 remaining species are not existing also during the Holocene. So the main difference between the Holocene and the other Pleistocene interglacials relies on the presence during Middle Pleistocene interglacial of a bunch of forest species and some of more open environment, which modern range is mainly Central and Southern European. During the other periods, the species out of range occurring are mainly species of more open habitats. So the following point is to know how it is, or is it possible to define a particular signature, malacological signature for each interglacial period. So on this table, I have removed all the species that are common to all the periods, and I have kept the remaining ones. In yellow are distinguished the extinct species, and in orange, the species that are absent today in the Somme Valley record. First, there are some species that occur only in one interglacial. Then you have species that occur in a limited time span. And again, what is showed by this table is that the main difference occurs between the Holocene and the signature of the other older interglacial periods. So finally, the last comment is a comparison with the current fauna of the Somme Valley. And this curve shows much higher amount of species in the current fauna registered on the modern survey. So what are these species, these missing species in the Quaternary? There are 41 of those. A little part, 14 of those are land snails, and among those land snails, most of them have been registered in the Quaternary in neighbor areas. So the real absence in the Quaternary relies mainly on this yellow group, and they are species of open ground and mainly of Mediterranean origin. But most part of the missing species in the Quaternary record are aquatic. And among these aquatics, there are only 4 of them that have been recently introduced, and only 3 that have been registered in Quaternary deposits in neighboring areas. So that means that there are different comments that can be done on this situation. One is that maybe the sampling can be questioned, because to perform paleo-environmental reconstruction, we generally favor terrestrial habitats rather than aquatic habitats. And also a question about preservation and identification, because they are really very difficult to identify. So the conclusion is that anthropogenic factors are only acting quite low on this fauna. So these are the important points, and I think I will let you to read them because I've been a little too long. So thank you very much.
Okay, so the fish are known to be today. They are known to be affected significantly by climate change, and they are an important food source that is currently under threat due to overfishing and habitat deterioration. The paleontological record suggests so far that the distribution range shifts that we observe today were also happened often in the past. So this is not something new. But the question is what limits, what makes them move? They do move. They move more than they do anything else, but they move very fast. But how fast and why? And when do they move and when do they come back? And when finally do they die? Because at some point they do die. In this study, this is the first record in the Eastern Mediterranean for the Pleistocene. There is nothing before that, and that's mainly due to the sediments in the deposits in Rhodes Island. The approach we used was to study fish otoliths, because fish otoliths, unlike the skeletal parts, they can be sampled through an outcrop, so we can watch the evolution of the fauna through time. And more similar to macrofossil analysis sampling process, it's very similar. So we can have continuous sampling. The resolution is not very high, but that's the best we can have. And the Rhodes Island is in the southeastern, I don't know if you can see, here is the southeastern part of the Hellenic Arc. It's affected by intense tectonics, and since the Pleistocene, it's undergoing anti-clockwise rotation and vertical movements that led to the lower middle Pleistocene deposits to rather deep water to be uplifted, and they are observed along the eastern part of the island over Mesozoic alpine basement that is severely eroded and faulted, and there are grabens formed. There were the deposits. The deposition took place during the Pleistocene, and for that reason also there are many changes in the faces, because each basin, each graben has not exactly the same faces. So there have been many distinctions of the faces, many studies done in these areas, and always changing. What we have studied is these parts, because during the lower middle Pleistocene, the area experienced a transgressive regressive cycle that started with Columbia Formation and followed by the Lindos Bay Formation, which was the deepest part, and then the Cape Archangelos, which is the regressive part. And these are the sampled sections, and the biostratigraphy was done by previous authors. We confirmed some of it, and these are the samples. So we identified the fish otoliths. Fish otoliths are sampled, they receive the sediment in the 250 micron sieve, handpicked and identified, and then we compare the distribution of the identified species with that in the Western Mediterranean, because there's differences today also between the Eastern and the Western Mediterranean fauna. And we use modern ecology data from the fish-based database. And then we separated the species. We used the 200-meter depth boundary, which is the aphotic zone, essentially. And it's also the boundary for surface water, intermediate water in the Mediterranean, to see how the different groups, the tropical, subtropical, temperate, and subpolar species that we identified were distributed, and how this changed through time between the surface and the intermediate waters. Overall, we identified 63 species, and we used their present-day depth distribution to estimate the palo depth for the Lindos Bay Formation and the Archangelos Formation, which is also it's a paper on its own, because it's valuable for the tectonic evolution of the area. I won't get into that. And this is I have some tables now about with the comparison of the Eastern Mediterranean. All the gray are the new occurrences. These have been reported before. These are the first time for the Eastern Mediterranean outside the Ionian Sea, because there have been many studies in the Ionian Sea, but nothing eastern than that. And these are Eastern Mediterranean, the whole. And all these data are from FishBase, are the modern data. And the very important is to note the anchovy. This is the anchovy, Graulis and Crassicolus, and Sardinella maderensis. And these are the otoliths of these two species are very fragile, so it's very important if we find them in the otolith record, it means they were there, and they were there in abundance. And also, both of them, they're small pelagic fish, and today they are abundant in upwelling regions. But Engraulis and Crassicolus requires to spawn, to lay the eggs, they need high temperature. So in the northern part of its distribution range, the spawning season is much shorter, whereas Sardinella maderensis moves along the western coast of Africa towards nursing sites that are more rich in nutrients. Also, this important here, the gadids, that these are the cods. We have Mediterranean cods, which is the Gadiculus argenteus. If you see here, this is the distribution in the Holocene. This is also the present-day distribution. When I say Holocene here, we don't have a record for historic times here. This is all based from the present day and some, very late historic. And upper Pleistocene, we don't have almost no record because we don't have deposits of that age. So we have the tropical Gadiculus labiatus, which starts from the Messinian but goes extinct sometime in the middle upper Pleistocene, or maybe in the Holocene. And we have the Gadiculus thori, which is a cold water species. It's a North Atlantic very cold water, and it came into the Mediterranean only at specific times. And then we have Gadiculus argenteus, of course, which also stayed from the Tortonian until today. Okay, sorry. These are some more. I won't go into every one of them. The one thing I want to note is that, like before, there are some species like Diaphostaningi that were observed from the Miocene, and they persisted through the Messinian salinity crisis, which was a very important event for the Mediterranean. They left the Mediterranean, and they came back after the crisis, but they disappeared in the late Pleistocene. And these are some, also Scopelopsis pleocenicus, which is a fossil species, it's an extinct species, also observed from the Miocene until the late Pleistocene. And another important presence is Protomictophilum arcticum, which is a very cold water species, that's North Atlantic also, and it entered the Mediterranean. We have only these records, and this one is not non-dated personal communication record from Ionian Sea. But it has been also found recently, last year, it's a recent paper on superficial sediments in the central Mediterranean, not continuous also. So this is a mesopelagic fish that came during cold periods, presumably, of course. And also another one is Chlorophthalmus agassizi, which is found, it's very abundant today in the Eastern Mediterranean only, and it appears whereas in the Western Mediterranean, the niche is occupied by Gadiculus argenteus that I mentioned before. A lot of new occurrences here because these deposits in roads are they're rather deeper water, but are still close to the coast. So we have coastal species, and this is rare to have otoliths from coastal species. We don't have such localities anywhere in the Mediterranean. And gobids also. So these are some typical photos, and I have the separation of the tropical, subtropical, temperate, and subpolar species that I found. This is also the present-day distribution of Sardinella madeirensis, the Madeiran sardine, that moves along here to find the nursing sites. The nursing sites are the sites the young grow to become juveniles and then move to the permanent sites to move along with the adults for fish. And essentially, that's what I make out from everything, that fish move, but their main trigger to move is to find the spawning sites and the nursing sites. So it's essentially a means to reproduce. If they can reproduce successfully, then that's the way they will follow the change. Because we have this in the present-day species, the present-day observations that fish follow change. It's a very common saying to show exactly how they move. Also, these are from the cods. And we have found that, okay, this here, yeah. Also, all three cods, the cold water, the Gadiculus thori, which is North Atlantic, Micromesistus potassius, not in the Eastern Mediterranean, just North Aegean, and the Gadiculus argenteus, they're all found together at 0.79 million ages in the roads. Then we move to the intermediate and deep water fish. These include, I forgot to say that before, the surface water. We included the fish that are epipelagic, and some mesopelagic that are shallow water, and essentially they move at night to be above 200 meters, close to the surface to feed. So they may be recording changes in the surface waters, whereas in the intermediate deep waters, we took everything from benthic and benthopelagic below 200 meters, and mesopelagic fish that migrate up to 200 meters at night. Okay, another important occurrence at 1.37 is Pagellus bugraveus, the sea bream, the red sea bream. We eat this, so it's a nice fish. And which appears together with Protomictophilum arcticum, the cold water mesopelagic. And then we plotted these, the distributions of the different tropical, subtropical, temperate, and subpolar species. Seems very chaotic, and I know, but it's a first attempt. And we're still searching how exactly to do and which fish to include, because we had to include some fish in two categories if they're, for example, some tropical and temperate their distribution. So, but I'm not sure exactly. We're figuring this out. But what we can notice is the very homogenizing here. And what we did again was try to see how some known suspects contribute to this. So, for instance, this is the three cold water gods that bring this the subpolar abundance high, and Protomictophum arcticum is observed here and here. So we picked these, the ones we already know from present data that are significant, to see how they're distributed, and what we note again is some seem to be important, like the cods. The Gadiculus tori is certainly important because we know it's a cold water and it actually competes with Gadiculus argenteus. That's why we think that here, for example, it enters the Mediterranean and tries to take the place of Gadiculus argenteus, so it lowers its abundance. So even though we observe them, because this is a very common, it's common even before the Pleistocene. It's a very common species. And it's actually, although it's a cold water, it's not so cold water, because it can be, if we consider that in the Pleistocene we have cold water called Gadiculus tori, then we have Gadiculus argenteus with the warm water species, the relatively warm water species. And then we have the Engraulus and Crassicolus to indicate some warm intervals. So my conclusions are we use the present-day distributions to estimate the depositional depths in the areas, we revise the paleogeographic distribution of 37 species. Also, what I found most important is that Miocene species disappeared in the late Pleistocene, and that for me is something unexpected, because I would expect a major shift after the Messinian salinity crisis and not so much in the uppermost Pleistocene. Thank you.
So today I'd like to talk to you about what stable nitrogen isotope ratios can tell us about past seabird activity in the Galapagos Islands. And before I get into the science, I just want to briefly acknowledge my colleagues, collaborators, and our sources of funding and support for this work. Okay, so why talk about seabirds? So seabirds are often referred to as sentinels of their ecosystems, or harbingers for changes in marine ecosystems. And that's because they sit at a relatively high trophic level, and so they're very sensitive to changes at lower trophic levels in their food web. So if the seabirds disappear from a given site, that does not portend well for the health of their ecosystem. And there have been many studies that have shown that seabird populations are declining. And this is a recent synthesis paper that suggests that this phenomenon is occurring worldwide. And so you can see from this downward trend in over 3,000 different seabird populations from around the world. And this is attributed to human impacts on the ocean, so overfishing, pollution. But seabirds can also respond to natural ocean atmosphere variability. And so a great example of that comes from this classic paper from Chavez et al. In 2003, talking about these biological regime shifts in the Pacific Basin. So as the Pacific decadal oscillation varies multi-decadally, there are these shifts in marine ecosystems across the entire Pacific Basin. And one of the most notable shifts is that anchovies are replaced by sardines at their trophic level off the coast of in the eastern equatorial Pacific, off the coast of South America. And so such impacts like this, they also ripple up and influence seabird populations. Okay, so I want to think about seabird variability through time today in response to these natural and human changes in marine ecosystems. And we're going to be doing that in the Galapagos archipelago. So the Galapagos are located off the coast of South America, about 1,000 kilometers to the west of South America. They're right on the equator. And where's the laser pointer here? So I'm sorry, this is really not appearing well. But so we're going to be focused on a lake sediment record from a small island called Henevesa, which is in the northern part of the Galapagos. And this is an uninhabited island, and that is also not appearing. Can we dim the lights? Can we dim the lights, perhaps? I don't know. Okay. So this island is uninhabited by people. Yeah. All right. So no people live on this island, but it is inhabited by thousands of seabirds. And, oh, okay. No, it's not. That's fine. So, but I'm interested in this lake because of the really beautiful laminated sediments that we find at the bottom of this hypersaline lake in the middle of this island. And I don't have time to get into the climate story here today, but these laminations can tell us something about past climate variability in the eastern equatorial Pacific. But I'm going to talk about the red-footed booby population that lives on this island. And so the great ornithologist Nelson estimated over 20,000 of these birds were nesting on this island in the 1960s. And these birds are everywhere. And they produce a lot of guano. So you'll have to take my word for it that this watershed is just coated in guano. Okay, so there are all these white streaks behind the birds in this image, which isn't appearing very well, but that's all guano. And so this guano eventually washes into the lake and contributes to really high productivity in this lake system. And so this is a hyper-eutrophic, hypersaline lake. And so the question becomes, is there some sort of geochemical or biological variable we can measure in this sediment to track guano inputs and seabird activity in this watershed? And that would be delta N15 values in lake sediment organic matter. So in other places around the world over the last decade or so, researchers have realized that guano inputs into these lakes elevates sediment delta N15 values. And that's because, essentially, delta N15 scales with trophic levels. So you are what you eat plus 3 to 5 per mil. And our seabirds are our top predators. They're sitting at the top of the food web here. All right, and so their guano is enriched in this heavier isotope of nitrogen, and that's making its way into the lake system. And so what should we expect for these guano delta N15 values? Well, they should more or less reflect the delta N15 values of the diet of these red-footed boobies. And these birds are primarily eating things like flying fish and squid, and they have been known to eat anchovies as well. So they're generalists as opposed to specialists. And so this is a plot of delta N15 values from 3 different sets of data. So as our no seabird end member, we've got some lake sediments from the Galapagos where we don't have a seabird colony in this watershed. All right, and so these are pretty typical values for your standard lake sediment, varying around 0. Here's the values from Henevesa guano samples, so guano samples taken from that lake watershed. They're pretty high, as you can see, and they more or less overlap values from the literature for these prey items of the red-footed booby. So that's encouraging. That's what we would expect. And then you can see the values from the Henevesa lake sediment over here, and they're pretty high. Okay, and so this is a result of that guano influx into the lake. You can also notice they overlap the guano values, but they are higher, and that's because other processes are influencing nitrogen isotopes as well. Namely, ammonia volatilization is one of the biggest things. And so as this guano sits on the landscape, the urea is turning into ammonia gas, and that has a pretty high fractionation factor. So there are other processes as well besides just guano input into the lake that are influencing delta N15. And that's been observed in other studies as well. But the main take-home point is that these really high values are a result of guano input and these birds in the lake. But as paleoscientists, of course, we're very interested in the variability. So what can that tell us about seabird activity through time? And so this is a plot of sediment delta N15 values for the last 400 years. And so we also have percent organic nitrogen and carbon and delta C13 values of bulk organic matter. And so the main point I want to make here for you is that when we have high delta N15 values in the sediments, we also have high nitrogen and high organic carbon percentages. And so that's a good indication that it is guano influx into the lake elevating the nitrogen isotope values and not other processes like, say, denitrification, which would lead to nitrogen loss. So we see a positive relationship between nitrogen and delta N15. And there's a very fascinating story with delta C13 and mangrove pollen. So this is pretty much a proxy for mangrove concentration around mangrove pollen concentration around the lake. And that may be connected to seabirds as well. All right, but let's focus in on the delta N15 time series. And so what will immediately pop out at you are these two abrupt shifts in delta N15. And so if we pass this time series through a regime detection algorithm, they do pop out as significant. And so we see these two abrupt shifts, one in the early 1800s and one in the 1960s. And the age model error is roughly interannual for the 1960s. We have a lead-210 chronology at the top of this core. And in the early 1800s, it's roughly decadal. Okay, so what is happening at this time? As a first-order hypothesis, we would say that these represent time periods when more birds are in this watershed and there's more guano getting into the lake. So was there anything happening to this larger Eastern Pacific ecosystem at these times? And so to look at that question, I want to show you some data from two marine sediment cores that are from off the coast of Peru here. And this is work by Gutierrez et al. And so the background map here is sea surface temperature, just to show us how this is a region of strong upwelling and thus cold sea surface temperatures and high productivity. All right, and so the G here is representing the location of Genovesa Island. All right, and so from these two sediment cores, they go back 700 years, and time here is on the y-axis. We're looking at a series of different indicators of marine productivity. And the main point here, the main point is that there's this abrupt shift in marine productivity around 1820, as stated in this marine sediment core, both these cores. And so we see that in diatom fluxes, as well as fish scales and fish bones and vertebrae counts. And so this is hypothesized to be due to an increase in upwelling and productivity as perhaps the subtropical high shifted towards the coast, or if you believe it, there was a northward shift of the intertropical convergence zone at the end of the Little Ice Age, driving these changes. So they are climatically driven. But what's fascinating to me is that what we see is the timing of this abrupt increase in marine productivity coincides with the timing of this abrupt increase in our Genovesa delta N15 record. And so that suggests that the birds are responding to this bottom-up increase in marine productivity in the eastern equatorial Pacific. And it's pretty straightforward to hypothesize why that would be. Perhaps it's because they have greater food supply, and so they are having greater nesting success. They're spending more time in the crater versus at sea because food is more abundant. Okay, but what about the latter shift here in the 1960s? That's more of a head-scratcher because there's really nothing going on in the early 1960s in terms of climatic shifts or anything like that. And this shift is happening around 1965 or the early 1960s. So what is going on here in this second shift towards higher delta N15 values? So what is happening is right at the time that there's this scaling up of the anchoveta fishery off the coast of South America. And so that's represented here by these anchoveta and sardine landings off the coast of South America, Peru and Ecuador, namely. And so what we see is this is not natural variability here. This is more and more fishing boats catching more and more fish. And that pretty much coincides with this abrupt increase in Genovesa delta N15 values. So that suggests the seabirds are somehow responding to this fishery intensification. So somehow that is impacting their ecosystem and rippling up and impacting the seabirds. And so there are several reasons for why that could be. It could be that prior to this fishery intensification, they're eating anchovies. These birds have been known to eat anchovies in other places, but now they primarily eat flying fish and squid. So perhaps it was a shift in diet. Another thing could be perhaps they're using these fishing boats as ways to track their prey more effectively, so they're doing better. Or they could be feeding on offal from these fishery boats. The other thing you're going to notice in this plot is that there's this increase in decadal variability. You can see all the ups and downs that are happening here. We also see that in our Genovesa delta N15 record. And the late part of the 20th century is really unique in that variance is very high relative to other parts of the record, and that's excluding those regime shifts. So there is this increase in variance in the isotope records, suggesting an increase in variance in the seabirds of this watershed in the late 20th century. And so what are the implications of that? Well, there could just be a natural increase in decadal variability, or these birds could suddenly be responding more sensitively to decadal variability. And that could happen if you have an exploited population, such as one of their prey items or the entire ecosystem. And so a population will become more susceptible to decadal variability at low population numbers. They have less of a buffer. But in sum, the main point I want to make is that it looks like the seabirds of Henevesa are responding to both that bottom-up increase in productivity in the early 1800s, but as well, they're also responding to this top-down impact of the fishery on their ecosystem in the 20th century. And I will leave it at that. Thank you.
You showed the strong increase in the fishing activity of Peru and Ecuador, but that's quite some distance where these fisheries took place in relation to your Galapagos record. So how did the Galapagos seabirds respond to the fisheries close to the South African continent?
Yeah, I mean, the idea would be that there's some impact on the entire ecosystem that's impacting the Galapagos as well as more coastal species. Or, the alternate hypothesis could be that decadal variability is actually natural, right? And so we're seeing it in the Galapagos and, a few hundred kilometers closer to the coast as well.
Hi. Do you see any link with the institution of the national park, which was around the 1960s?
That's a very good question. I don't know. I don't have a great answer to that, but yeah, that's something to think about.
Because you see the increase in variability. I mean, Genovese is not really populated, but the ban of the fishing, it's mainly coincident with the 1960 period. So
yeah, that's intriguing. Yeah. So I mean, no one's ever living on that island, but definitely there'd be more fishing boats.
But yeah, they come from Santa Cruz and Cristobal.
Yeah. Thank you. That's a good point.
Well, now I'm going to take you to the deep sea. Slowly over the session we moved from the forest to the coast at least, and now we're going to the deep sea. And even there we have strong environmental changes that affect the ecosystems and cause some trouble down there. And I hope I can show you how these ecosystems can look like down there in the deep sea. They're really fascinating ecosystems and they're quite diverse, and I will, thank you, tell you one story about a case study, in a way, about such environmental changes and their impact on the cold water coral ecosystems across the Strait of Gibraltar. But maybe before I start, I say a few general words about the cold water corals. We are looking into the framework forming ones, which make these nice frames that also give space for quite some other species. But first of all, the cold water corals occur almost worldwide. We know them from northern Norway down to the Drake Passage, from all ocean basins. And so that's a much wider distribution than we know from the tropical corals, which everybody knows that occur in the shallow, warm tropical waters. But these cold water corals, they occur almost everywhere. And as I already pointed on, and as you can also see here with the frameworks they are forming, they create lots of ecological niches, and so they are supporting highly diverse deep-sea ecosystem, which in terms of species numbers are not on the same level as the tropical coral reefs, but they are coming very close to it. So these are very biodiversity hotspots in the deep sea. And the corals are instrumental in creating impressive seabed features, which we call coral mounds, that can also be used and exploited as paleoarchives, what we do. And these structures, these coral mounds, they can get up to more than 300 meters in height. So that's really impressive structures. Well, the corals obviously are sessile, that means immobile benthic filter feeders. And when we talk about move, adapt, or die, well, the move we already can skip here at this point. And they do have a quite turbulent regional histories in several parts of the oceans throughout the Quaternary climatic cycles. And I will take you now to the Straits of Gibraltar, connecting the Atlantic Ocean with the Mediterranean. and take you to two cold water coral sites. One is the Moroccan Atlantic margin site, where today we only find fossil corals, and the other one is in the Ivory Sea in the Western Med, where we have also the living corals these days. And that already tells you a story that there are differences in the terms of vitality of the corals, and that's something we try to follow into the past. And we did that. We collected surface samples and short sediment cores from these sites. But 2.5 years ago, we were able to go there with this new instrument, the Bremen seafloor drill rig, to obtain up to 70-meter-long sediment records from these sites. And today I'm going to show you the first results from these records. Just a brief introduction. That's a map of this western site in the Atlantic. There are some interesting features you see up here, but that's not coral mounds, that are mud volcanoes. I'm afraid you cannot see the coral mounds. That's all this little dots around here, but there are hundreds of them. We mapped them, and there are more than 600 mounds in this relatively small area. On average, they are about 20 meter high and occur in water depths between 700 and 900 meters. It's a little bit different on the other side in the Mediterranean Sea. There are some very strange features, these long winding ridges here and here. Then we have the circular mounds over here, and these hard to see, almost blank, almost draped by sediments, ridge-like structures over here. But all of these structures, these mounds here and these very different types here, they are all made up of a mixture of cold water corals and sediment. So they're basically the corals are instrumental in creating these features. And in this particular ridge here, we drilled down to 70 meters, and 70 meters below the seafloor, it was still cold water coral. So the entire ridge is made up of these creatures. And at both sides, we have the same dominating species. That's Lopheria pertusa. That's the most prominent one we have in the Atlantic, and that's also then the most common here at these two sites. Let me take you to this specific site here to show you how these sites look like. That is the Wulle Mound. That's a parasound section through it. So that's the seafloor here. You see the coral mound. It sticks up to 20 meters above the seafloor, but it's also a lower part buried within the sediment, so it has a longer history back in time. And the entire height of the structure is 35 meters, and we drilled through it, and we found corals in this upper part and then the normal sediments below. That's how these sediments look like. The corals are embedded in a hemipelagic sediment matrix. And then to get an idea about the history, we picked out the coral fragments, we cleaned them carefully and dated them by uranium thorium to get a precise chronology on these corals. That are the first results. That's now, again, the western part, the Atlantic part. And we did the datings on two species, mainly Lopheria pertusa, but also some on Madrepora oculata, which usually occur together. And you see here the data, that is the core depth, that is the age, and this violet color you have the interglacials indicated. And you see that we have a predominant occurrence of these cold water corals during the glacials. That's not a black and white pattern. So it's really a predominant feature. You see here is also an interglacial one, and probably also the very early Holocene. There are still some. You see some more data from the older records in a minute. But as I said, there's a predominant glacial occurrence of these corals on the Atlantic side of Morocco. Now, when we go to the Mediterranean side, it looks a little bit different. Then we have a predominant occurrence in interglacial periods, and interesting here in marine isotope stage 9, stage 7, there's few data, or almost this, you will see in a minute, there are one of two data here, but hardly any data in stage 5. At the onset of the Holocene, we have some data here from the shallower records. We have more data, and you see the onset of the Holocene is in 10-meter sediment depth. So over the Holocene, we acclimated 10 meters of sediments bearing these cold water coral fragments, and that's the average. So there are periods, especially at the very early Holocene, when we got 5 meters of record deposited within 1,000 years. So that's a very fast coral carbonate accumulation, if you like, from a geological point of view, but it also tells you from a biological point of view that the corals were really thriving at this time. So, but then we have what I as already mentioned in the title, we have this kind of seesaw pattern, and that's a bit more amplified, or more clearly shown here. And here you also see some older data. That's all the small dots from short cores and surface samples, and so far from the Mediterranean we only had Holocene ages, but now we have, and we set up the hypothesis that they are limited to interglacial sections there, and basically in a general sense we have the interglacial occurrence in the Mediterranean and the more glacial occurrence in the western, in the Gulf of Cadiz of Morocco then. And that's a more or less continuous pattern over the seesaw pattern over the last 300,000 years. That's the record we have so far. Now, we can say that, and to come back, now it's plotted against time here, but then between these dating points we have hiatuses. So there's no more coral proliferation and no more deposition of these coral fragments, and that means also that the finer sediments are no longer captured by the coral framework, and so we really have hiatuses, which we see in different cores in different settings. So that's always in both settings. It's a really regional signal we see. So we have these several on and offsets of coral growth in both regions with this kind of seesaw pattern. And so we already skipped the move. Adaptation obviously also does not really work here. And that might lead to the conclusion that the rates of environmental change have been probably too fast for the corals to adapt. Well, forcing, we did some work on the forcing factors, so we have the coral records telling us when the corals lived and when they were absent. But we used the normal paleoceanographic approach to learn about the environmental factors at these sites. And these are very, very clear in telling us that basically it's always a kind of regional pattern that affects the corals, but it's mainly the food supply that is controlling the proliferation of the corals. And, well, can you really adapt to no food? That's a question marine biologists is asking to this topic. That's not really adaptation. We know they have some adaptation capability, the cold water corals, in terms of temperature or oxygen conditions, but to adapt to having no food is probably very difficult also for very adaptive people like we are, for instance. Well, the seesaw timing we see at this site is something we also see at different sites. That is an overview about what we know about these long-term records of the cold water corals. The map in the back, all the blue shadings shows you where corals have been found. Well, that's from 2006. We know a bit more by now. And then the larger dots show where we have these coral mounds and where we get these paleo records from these sites. And there are some marked in yellow here where we have predominant glacial occurrence of the cold water corals, and the red ones where we have a predominant interglacial occurrence of the corals. For the blue sites we have no information at all, and we also have a very few sites where it seems they occur continuously. So the seesaw pattern I just described for the Mediterranean versus the Gulf of Cadiz is also the same we see from the Gulf of Cadiz looking northward to the Irish and the Norwegian margin, where the most prolific cold water corals occur these days. So it seems that at least for the entire northeast Atlantic, especially this area of Morocco has been a kind of glacial refuge for the cold water corals. Also here on the northwest Atlantic side, we at least know two sites where they appear to be limited to interglacial conditions. So well, we are still at the beginning learning about these coral mounds, but there's definitely a very interesting story in it. And we know at least that on a regional scale, the constraints, there are strong time or basically climate control on the coral development. Putting that all together, we see that there is this seesaw pattern in the occurrence of cold water corals across the Strait of Gibraltar. And to what we know from our paleostratigraphic record, it seems it's controlled by regional oceanographic changes, at the end controlling the food supply to the cold water corals. When we talk about these repeated regional extinctions, when we can say that might be a denial of any adaptive capacities of the cold water corals towards environmental changes on the respective timescales these occur. But again, is food shortage really something you can adapt to? Interesting part is that when we have the cold water corals stepping out because they do not get sufficient food anymore, then we lose the entire ecosystem. In some of the North Atlantic sites, we have 5,000 species associated with these cold water coral ecosystems, but when the engineer is gone, the ecosystem is gone. So that really has wide-ranging consequences. We can combine the coral fossil record with the paleo-environmental reconstructions and learn then about the forcing factors that control the corals, and basically that also control the ecosystem development then. And that brings me to the still open thrilling question we are dealing with. It's somehow easy to see how the corals get lost when they do not have enough food any longer, but when conditions get better again, they are immediately there everywhere. So that's something we try to understand these days. Thank you very much.
Do you have any idea what kind of oceanographic changes these are that deplete the food supply?
Yes, I think it's a kind of reorganization of the water column structure. We find them mostly at places where we have water mass boundaries. And these water mass boundaries are creating more energy through all the variability between the water masses. We have the internal waves and internal tides. And this energy basically is supporting the food supply by moving food particles to the corals. The corals they cannot move, so the food has to come to the corals. And the more energy we have in the water column, the more water movement, the more food the corals get.
Yes, thanks for giving me the opportunity to speak here about my work with ancient DNA, which is why there has been this confusion, on large species, turnovers, and vegetation change in the Arctic boreal tree line ecotone during the Holocene. So to start off with, here's a picture of the tree line in Siberia. I think this is it. Well, you see this. It's a very large ecosystem boundary. It's an important boundary between forest and tundra. It's a wide ecotone with gradual change. So here, I'm not sure if you see it, but these are pictures taken from a helicopter where you kind of see different types of vegetation where with open light northern taiga. There will be other pictures if you don't see it here, going over to where there are hardly any trees and then really where there are really basically no trees left. And this whole ecotone is projected to change with climate warming. And there will be associated albedo changes and, of course, biodiversity changes, which I, as a biologist, am particularly interested in. The situation is not as simple as you might think. It's not just trees going up north, but it is actually more complicated. And we just had a paper now accepted in ecology showing that there are dissimilar responses in different parts of the tree line, that it's not all reacting identically. And so the situation is complicated. And why is that so? What is actually the thing? So first of all, it's made up of larch in this area, monospecific stands only of larch, of larix. But, and that's the thing, not only monospecific, you might think, and as if you look at pollen, you can only see larch, but there are actually 3 different species, or also more if you go in other areas, of larch forming this tree line. So we have Larix sibirica, in this part here, then Larix gmelini and Larix kayandere in the whole part in Siberia. And if we think, so this is the modern limit of larynx and the projected limit of larynx. This is a bit of an old figure, but a classical figure showing what we think might happen. There will also be a species distribution change of large species themselves. And it is a very remote area. We don't have a lot of empirical data on what is happening at the moment or what has been happening in the past. Now, these large species are actually distinguishable both by their mitochondrial haplotypes. There are large areas of hybridization, that I must say. So they hybridize, especially here in this area. But there are haplotypes, so different types of mitochondria that seem to be diagnostic for the different species. So in the area of Larix gmelini, I will put it this way. In the area of Larix gmelini, of pure Larix gmelini, the trees have a certain haplotype. And in the area of Larix sibirica, the trees have a different haplotype. And in this hybrid area, you see different haplotypes in different trees if you look at modern populations. And they also have ecological differences. So Larix gmelini is generally considered to be the more hardy species. For example, enduring thawing depths of 10 to 20 centimeters, while Larix sibirica needs 100 centimeters of thawing depth. And also, endured winter temperatures are lower for Larix gmelini. So in general, you would think that, okay, Larix gmelini is in the more continental area, so it's more to the northeast, than Larix sibirica, which is also what we see as a large picture. And we tried to now look at this situation in this hybrid region or this boundary region more closely to see what's actually happening. And to do that, we first of all, so this is what I did with ancient DNA metabarcoding and pollen to get an overall view of the vegetation and vegetation changes. And also, I looked at this particular marker, this mitochondrial marker, to see what was happening there. And in parallel, we ran a model, which I will introduce in a few minutes, incorporating the two species. So we have samples from expeditions covering the tree line ecotone of this region with a helicopter expedition, sampling of lakes, and vegetation mapping. And what we did is we sampled lots of lakes, surface sediments, and sediment cores. And this, for those of you who were in the ancient DNA session, this is quite familiar. DNA extraction, PCR. This is not so familiar. Most of the talks were only targeting vascular plants. And here I also used a Larix-specific marker for this mitochondrial haplotype. And then processed the whole thing with Illumina sequencing and looked at it. So does the DNA in this area actually show us the vegetation? We first ran a test on surface sediments to see if we are actually seeing what we expect to see. And we see that the DNA metabarcoding, first of all, gives us a high diversity in taxa, 114 over this whole transect. So these are the surface sediments. The pollen, less diversity, and vegetation surveys, admittedly from plots that were not directly around the lake, but close to the lake, even less. Those were not very thorough, maybe you could say. But it was what people usually do when they go to the field and they just have a limited amount of time. What we see is a good fit. And we see that all methods can identify the tundra composition, at least compared to all the rest. So the single tree tundra, the forest tundra, or the light northern taiga is really different. So when there are trees, we see them, or we don't see them. So let's say we're at least not seeing something that we would be seeing differently if we're looking at the pollen. If it's true or not, that is another matter of debate. This also goes through the cores. In the cores, the picture is extremely similar between the DNA metabarcoding and the pollen. So I plotted them on top of each other, and these are reads. This, again, was a matter of debate in the other session. These are reads showing that we see very, very similar trends between the pollen of Larix and the pollen of Larix and the DNA reads of Larix. We see that both in the absolute counts, and also if we run on the complete DNA data or the pollen data, PCA, and then we look at the trends in the whole PCA, it's also extremely similar. And so we think there again, we see a similar picture. And we see here a fluctuation of the forest cover, whereas, and this was also published by Juliane Klemm, 2015 or 16, the pollen data, a decline in forest here from around 7,000 years ago to now. And this pollen data is from this study. So what do we see in the haplotypes, actually, of this area? We see something that I, at least, was not expecting. So this is the overall picture. Over here we have Larix mielini. Over here we have Larix sibirica. I thought when I was waiting for the data, I thought we would probably see, over here will be all Larix mielini haplotype, and over here, well, maybe some mixture of this Larix sibirica, but well, we'll see if we actually hit it. But what we saw was that this core started off with Larix sibirica down in the early Holocene, and then there seems to have been a really a complete shift. So these ones are kind of mixed haplotypes, which this is per definition, I defined it, if 10% or more of the reads are of one or the other. And if it's below that, I said, well, it's really dominated by one of the two types. So in this more southern core, we seem to have a real turnover at a certain point. Whereas in the northern core, and that I find particularly interesting, we have mixed haplotypes, nearly until very, very recently. Up here we have very little Larix, as you also saw in the overall vegetation. And here we find only the one haplotype, which is not so surprising. But we see a mixed signal throughout the core. And this is also repeated when you look at the surface sediments. Well, not quite. What we see there in the surface sediments is even more striking, that the two northernmost lakes that we looked at, they had this Larix sibirica signal, whereas the others seem to have the Larix mielini signal in surface sediments, except one with a mixed signal. So this was even more striking in a way. And then we ran a model to see what would happen if we take the ecological parameters of these two large species and see through time and in space how they would distribute themselves. So this is an individual-based spatially explicit model. If you want to discuss this, maybe afterwards, and I also have to say I am not the modeler. This was published last year by Stefan Kruse. And what it does, so it goes in cycles of 1 year, always through many, many years, 500 years or 1,000 years, taking the environment, first of all, as one factor, abiotic, biotic, and then the different species that you insert. So with different parameters for growth, seed production, seed dispersal, establishment, mortality and aging, and then goes round through time. So we did that with exactly these different parameters that I showed you. That was the master thesis of Nadia Khat, to see what would happen. And what happens is the following. Across geographical scales and for current conditions, we have a really good fit of how the species distribute, except for one thing, or a couple of things, but one that I would really like to point out. So to understand this figure, we have different insertion of the species in different stages of this model run, right? It's a long time, 150 years of stabilization, and 500 years of running to see what's actually there. So in this point, for example, or in this point, you would insert only Larix sibirica all throughout the model run. And in this point, down here, only Larix mielini all throughout, so basically you can't have anything else. And that's, of course, what you see in all these different simulations. But you see, and that's very interesting, that especially in this northern plot, if you see different little, you see these mixed populations with certain conditions, inserting the species in different ways. So you do not always have a serious a turnover or a dominance of the Larix mielini in the north. And then we ran that temporally, and there, that's also quite striking, we see that in the southern plot, so these are exactly the two plots from which we have the cores. In the southern plot, we see Larix mielini, as we kind of, similar to what we see in the core, and in the northern plot, we see at some point, we see Larix sibirica appearing. And we see this even more with, this is just a simulated plot, Khatanga North, and so you see a population drop, and then this other type comes in. So overall, we have a quite interesting fit between what we see empirically and what we would also model for how this whole thing behaves. And so what we think we're seeing is basically the competition between the species. So the boundaries, the precise boundary, not the overall picture, but the precise boundary are set by competition. And the tree line ecotone, that's the other really interesting thing for us, is that it actually harbors the different ecotypes. And depending on how the climate changes now and how fast the whole thing goes, we will see how they will react. And overall, of course, the dynamics of change are much more complicated, and it's not just larches going up and to the north, but there will be different types reacting differently and competing with each other, which will affect the speed and the whole process of how this is going to happen. Yeah, that's my summary slide, and so I think I'm also done. Some acknowledgements briefly to people who have been working on this project, and the Alfred Wegener Institute and the DFG, who's funding me at the moment. Okay, well, thank you very much.
Can you do any forward modelling, like predictive modelling,
yes,
for how it will happen?
Yes, we can. And I'm not quite sure, I don't really remember how it is in Stefan's paper, but of course not for the two species, we haven't done that yet, but in general, yes. And yeah, of course he did. And the whole, when he described the model, that was of course one of the, yes. But we haven't done it for the two species.